did you do a statistical analysis on your results?
I'm sorry?
Did you do a statistical analysis on your results?
What do you mean there's 100% correlation? Let's be accurate. On one patient, only 1 in 700, she was like, I definitely have it, my test.
What was the definition of the COVID illness?
What was the definition? Well, I'm talking about a PCR test.
No, you said it was 100% correlation with having COVID. What's
No,
that?
100% correlation with a PCR positive test and an antibody test. 100% correlation.
It's impossible to really know the answer because we don't really know what they're detecting. So, I mean, if you had 700 tests and they gave identical results every time, then it means they're correlated with each other. But it doesn't tell you anything about what they're measuring.
But there's got to be something. There's something that they're accurately measuring.
Why does it have to be accurate?
Because it's statistically impossible that all these tests would correlate otherwise. Explain. If you came to me and you said,
So I don't think really we're able to explain. If you want to give me the data set to look at and give me your written up procedures of exactly how you did it, I'd be happy to look at it. But the thing is, the tests are not meaningful.
Right.
We don't know what they're actually assessing, and there's no clear clinical correlation. So even if they correlate, that's fine, but it still doesn't mean anything in terms of clinical significance.
but
So
it's
why don't we get the
No, we have to
yeah.
move on.
So I have a few different questions, if you don't mind.
We have a one-question limit. After this, if we stick around, of course, but I want to make sure that everyone gets an opportunity to ask.
Quick question.
Events, I created a sign with Carrie Morris's quotes, and these are his quotes from the videos that are available on BitChute, etc. That the PCR test does not tell you that you're sick.
Yes.
That's as simple as that.
Or the judge.
You know, Kerry Mullis, when he came to the conclusion that there was no HIV had anything to do with any disease called AIDS. And he didn't say anything for about 12 years. And this is such an interesting point that he made, because he said he couldn't but believe that there must be some wise men out there, and if he could only talk to them, they would tell him what he missed, because he's just basically a surfer bum. And so finally, he gets a Nobel Prize in chemistry for discovering the manufacturing tool, which is called PCR test, and he gets invited to a dinner of Nobel Prize winners. And this is sort of the question that we all have. There must be some wise man out there who can tell us. So Carrie is now one of the wise men. And he goes to a dinner with the other wise men. And he goes up to Luc Montagnier, who was the co-discoverer, so to speak, of HIV, and said, finally, Luc, tell me a paper that shows that HIV exists and is the cause of AIDS. And he gave him a paper. And Carrie, who is not just a surfer bum, said, I know that paper doesn't even have anything to do with HIV or AIDS. And Montagnier walked away and would refuse to speak to him. At that point, a light went off, which is the same light that's about to go off with all of you. There is no wise men out there. There are no wise men who can say, this is the way it goes. That's a model which is dead. And he was fortunate enough to be in a room with all of them, and the emperor had no clothes.
This is, I just want to add something, because I think this actually also speaks to your question about how can we be prepared for the future. And I think that, it's kind of going backward, but you could look at it as a philosophical change, that we need to return or go forward back to an era where we take responsibility for ourselves. It's our responsibility to maintain health, to learn what's going on so that we can make the right decisions. And we have been, really reared and conditioned to give out that agency to designated experts and authority figures. And that's why we're looking for that wise man or the so-called grandiose other who will come in and rescue us, save the day, right? But that's just ain't gonna happen. It's our responsibility, right? That's why, even though I'm a psychiatrist, I've become a virologist of sorts, right? Because necessity compels that in this moment.
And by the way, I did a admittedly sarcastic thing on antibodies. And I will just redo that in a second. Because antibody, so here's the way we're told. You make you get caught, you catch a virus, the virus lives in you for 2 weeks, that's the amount of time you can find a PCR test. Then you make IgM, which is a non-specific antibody, which also lasts for 2 weeks. You can see the spike. After 4 weeks, then you start making IgG antibodies, and that's specific for that particular virus and lasts the rest of your life. That is, of course, is because both viruses and antibodies understand the concept of weeks. So if you go to a different culture that has an 8-day week, it would be the virus lasts for 16 days and the IgM for 16 days, etc. So an example of this is you get infected with mumps, and then because you make this sequence of antibodies, you're immune for the rest of your life with mumps. So that's one example. Now, if you take measles, the same thing happens. You get infected with the virus, then you make IgM antibodies, and then you make IgG, which is specific to measles, and then you're immune for the rest of your life against the measles virus, except let's not forget about that one because we know the measles virus doesn't exist. So then you have chicken pox, where you do the same thing, and then because you have IgG antibodies, you're immune for life against chicken pox, except if you get shingles, which means you have a different variation of chicken pox. So somehow the immune for life antibodies didn't work. And then on the other hand, if you have symptoms of AIDS and you have positive antibodies for HIV, then it's very clear that antibodies mean you're infected with a deadly virus and you have no immunity. And if you really understood that, people would start laughing. And we have another example of Hep C, which is diagnosed by the presence of antibodies. So it's very clear that antibodies mean you're infected with a deadly virus, and that's going to make you sick, unlike mumps, where the antibodies mean you're immune for life. Now, the flu virus, which is a very wily virus, in fact, it has stock in the vaccine manufacturers, that mutates every year so that the antibodies only last for a year, meaning you have to get a new flu vaccine every year. And then I put in the book a quote from the head of infectious disease immunology at Wake Forest about antibodies to the coronavirus. And he said very specifically, having antibodies to the coronavirus means you were either sick or you weren't sick. You either have the virus or you didn't have the virus. You either are immune or you're not immune. And if anybody just understood what I just said, congratulations. And I have a gift certificate to the local funny farm. Antibodies mean nothing. Yeah.
So I watched your video about the mRNA vacations, and you said that they do change your DNA because of all the cells have reverse transcriptase. And then I saw the material from the vacation manufacturers, and they say that the RNA directly affects the ribosomes. It bypasses the DNA and then to cause the proteins to be constructed. So I was going to ask you if that explanation makes sense. It's still genetic modification, but it doesn't modify the DNA. But now after what I've heard today, it sounds like you don't think genetic material can enter cells at all. So so what do these so-called vaccines actually do? Do we have any idea what they do? Right?
Well, we don't really know exactly what this genetic technology does, other than it says that we're going to make whatever is coded in that gene, but that's not been demonstrated, right? They don't have cellular cultures where they're using this and then they can measure how much of that protein is made or anything, not that I'm aware of. But in terms of RNA going in and out of the nucleus, this happens all the time, right? Because so in order for DNA to
So
make
the
a protein,
RNA isn't like the string that won't fit in the thimble, it actually does go into the nucleus?
yeah, so that's DNA. So we didn't really differentiate this a lot, but basically DNA is the big long strands that make up the chromosome, and if it wants to make something, it transcribes in the code into RNA. And those RNAs are not as big as long, anywhere near as long as a chromosome, but some of them can be quite long. Those RNAs, there are many, many different types, and they have many, many different, they do a lot of regulatory functions of the cell, they regulate gene expression, they do all kinds of stuff, okay? They respond to illness. But the RNA is made in the nucleus, and it crosses out of the nucleus, because the proteins are made in the cell membrane. And then RNA also goes back into the nucleus, and they can undergo reverse transcription. So RNA seems to be able to travel across the nuclear membrane by some unseen mechanism. Now, whether the mRNA from this gene therapy technology actually goes into the nucleus, we can't really say for sure because we don't have an experiment that shows that. Right? But it does stand to reason, based on our knowledge of how they report the general properties of the cell and the genetic constituents, that there's a good chance that it would. And that's how the virus would work. Like, it's actually the virus model in itself, because it would come in with its genetic material and it would get into the nucleus and integrate its material into the nucleus and then start making mRNAs to make its properties. So it sort of, really fits what their model is. But we don't know, if it's actually true or what actually happened 100%, but according to their model, I think that's the logical conclusion.
I can also tell you that having read, anybody who's really interested in biology and science and all this, I'm almost to the point where if you're a doctor or scientist and you haven't read Harold Hillman in its entirety, like I did the last month, then I don't want to hear from him. Because he has 100 pages of showing that it can't possibly be ribosomes, which then makes the whole thing crazy. Like, you start dissecting this down to the details and it becomes a morass. And so it's really hard to have any clarity on exactly what is happening with this stuff.
And then a lot of people I know who don't believe that the virus is contagious, they think that people who get the injection are then contagious, and I have to argue with these people all the time. There's no way.
That's the kind of, mental traps that you get into if you don't just look at this existence of the virus in the first place, because all that's not true, or you wouldn't even think about taking a vaccine if you knew there was no virus to get a vaccine for. Mm-hmm. So you wouldn't have to worry about the safety of it. Like, people ask me, is it safe? I'm like, that's the wrong question. Yeah. Is it effective? Is it needed? Is it real? Is it real? What they say it is, right? It's none of those things.
So, yeah.
I want to go back to, how do they justify any of this next generation technology? How do they justify any of it without using DNA primers? I'm kind of mind blown.
They say that it's unbiased because they don't use primers. In other words, they take all of the DNA or RNA in the sample and amplify it, and then the computer selects out, through that process I outlined, what is relevant or not, right? So they call that an unbiased process. So you see, there's, like, a lot of misuse of terminology, or, you can understand it in that sense, but the whole overall process is extremely biased.
Here's another way to answer that, which is what I came to. in working with this. And this came out of a conversation I had with a guy named Naveen Jain, who was, he's a billionaire, founder of this company called Viome. And it really is the crux of the issue here. So he had this program, so he actually got the guy who invented Watson for IBM to work on his project, and they were going to basically take stool samples and figure out what bacteria and viruses and fungus were in your stool, and then correlate that with the published research and tell you what to eat so you would have optimal health. So that was his, and for a while they became very successful, and then I think they folded. And I had a conversation because Naveen wanted me to use his stuff and maybe even talk about it or endorse it or something. And we eventually got to the crux of it. He insisted that this way of matching the microbiome to the food was completely objective. Completely objective. And I said to him at the end, but didn't somebody do a study and put that in the computer, and that's how you knew that broccoli is good for somebody with Acinetobacter 2% instead of 1%? And he said, no, the computer figured that out. And at that point, we were stuck. Because even the questions about, is it the same composition of your stool today as it is tomorrow after you eat yogurt? No, it's completely objective because we didn't do anything, the computer did it. That's the problem. And so once you get to that point, there's no argument. They say, no, this is completely unbiased because we didn't do anything, the computer made the sequence. And if you ask, like, who programmed the computer? Or how did the computer figure it out? No, that's the unaskable question. That's where you get stuck. That's the problem. It's fundamentally a belief in the primacy of transhumanism. We are simply just computers. That's the answer to this question. That's why we believe.
I think I know the answer. It's derivatives.
Derivatives.
Bad joke.
Derivatives.
Stop it. It's a little crash. Jesus, is that really caused by a toxin like
I think you missed, you talked about it for 10 minutes earlier.
sorry.
Let's go on.
I just wanted to answer, well, kind of respond to that, but second question about what do we do next? Because I truly am a strong believer in, it is like Andy was saying, it is us. I've met many business owners that have never closed, and their business is booming because they've never closed, and people that are awake gravitate to them. There is going to be this, March 1st is small business owners across the country are coming together and declaring the pandemic is over. No more masks, no more required. Yeah, I mean, if a person wants to come there with a mask, they're not going to not let you in. But they're going to take down all those signs. Over 3,000 business owners are coming together on the 1st of March and saying this path is over. And it starts with us. Us individually, each one of us has to be awake and walk and live that truth, and then we can share it with others. You don't have to fight with others. Just share it. If they don't want to listen, then fine. But you live your truth, each one of us. It's not going to come from the government. It's not going to come from, science, the fake science is not going to come from Fauci. It's not going to come from Bill Gates.
So I have an answer to your question. How is this going to resolve when we elect you to be the minister of I think we have just a few more.
Can I ask a 5G question?
Yeah.
I live on I work on the south end of Syracuse. They have it's one of the cities that has some 5G going right now. My work is probably within a quarter mile or so. So I just want to ask your opinion, like, how would you look at that if I should I be trying really, really hard to work from home, or am I? Yeah, I know there's probably not a real clear yes or no answer on 5G, but do you have any opinion on that?
I mean, the model I'm working with is, when you get right down to it, that we're made of organized, coherent, or sometimes called multipolar, high water with a bunch of stuff in it that has a membrane that's organized by the energies of life. And 5G frequencies are not the energies of life.
That's right.
And how bad it is, who's using it, where it is, it's really, it's a huge morass. But I can tell you, from everything that I've seen, that's not something that organizes us to be the kind of spiritually evolving human beings that we want to be. So there's lots of different ways of mitigating it. I've had interviews and interviews. I mean, who knows? We're all working on this to try to see what we can do. If anything, the best is to get rid of it and create a different world.
What are some mitigating methods?
I mean, I have, go watch my podcast. I have interviews and interviews about that. But it's really a hard subject to wrap your brain around, because it's the same thing with why aren't we studying what happened to that hole? It's like individual people trying to get the money to study what the biological effects are. And frankly, some of the 5G researchers, in my opinion, are using the old paradigm, like it affects calcium-gated pumps and voltage things in the membrane. But the fact of the matter is, you can't prove the membrane, you can't prove a calcium-gated voltage, something in the membrane anyways. And so it's just, it's a morass.
Protect yourself.
I think a morass is my head. Morass.
And that appearance that being near someone who has symptoms you could catch, could it be related to the coherence of the water in the bodies that are like creating the same structures? So to
Yes,
speak?
it could be, but I think we also run a serious risk of overdoing that. Mostly it's common toxins and just maturation process.
So I have, in your nice picture of the mRNA vaccine, it said mRNA is made with instructions to make viral protein. Is it possible to explain that? I didn't get. Is that like they went into the bat and took?
No. It's from the in silico genome that they created using a computer. They say that there are certain genes in there, and there's one gene with a spike protein. So they just basically took the letters from that and popped it into the gene therapy vaccine. So it's supposed to basically whatever's coded in that computer takes a base and polymerizes it, adds one base at a time, makes a chain. It's just the same thing that happens in your cells, but they have a laboratory procedure. It's just chemical reactions. You join one base at a time onto the chain, just like if you were making a necklace out of paper clips.
It's just making bleeding stuff, and you got to keep it around for a while. So that's what they figured out how to do.
So I just want to get perspective on it. If this room was a cell, how big is a virus? The second part of the question is, have they ever isolated the virus?
No, except bacteriophages and so-called giant viruses. Those are the only two that have been, as Andy showed you. There are bacteriophages. We used to think they ate the bacteria, and now we think they're like, if you stress a bacteria, like in a laboratory, it goes into a protective mode, which has different, composition and shape and intricate pattern depending on, I think, what you started with and how you stressed it.
Right.
So you get a flying saucer if you put arsenic in it, and you get and it comes from a strap or whatever. And so those have been isolated. And the importance of that is that proves that you can isolate something of this size.
Well, how big is it?
Same size, 100 to 200 nanometers.
Well,
And that's a
how big
billion,
is a cell?
billions of a meter. The cell is micrometer scale.
Micro,
Billions of
nano, so
cells.
it's 1,000 times smaller than a cell.
Yes.
But the point of it is the argument that we hear a lot that you can't isolate something this small is completely false. It's completely negated by the fact that that's exactly what the research on bacteriophages did for 20 years. And that's what Stefan Lenka did. He isolated a so-called giant virus, which is just a little bit bigger than normal virus, from a sea creature. And then he found out, hey, the ones that have this so-called virus are healthy and the ones that don't aren't. And something's wrong here, and I better not say anything for 12 years because there must be some wise guy out there who knows why I'm wrong. And then he figured out that he wasn't wrong. There are these little particles in stuff, and you can isolate them and characterize their whole genome just like.
She survived rabies. Yeah, that's what.
Did she really have it, or
That's
did
what I thought too.
she get the vaccines?
I don't think she actually had it. I think it's one of those things where she got bit by a bat and then
She
she's
didn't actually have it.
right.
But here's the thinking, Trump.
There's no person I've ever.
Here's the trouble and where we get into. People have the experience of, I got bit by a dog or I got bit by a tick and then I didn't do well. Right? That's a human experience. Now, then they say, therefore it must be a spirochete. Now, that's not a human experience. That is a laboratory evaluation. Because you, at that point, you know, if you get bit by a snake and you die, there could be a lot of possible things in that snake venom that made you sick. In fact, I got into this with Mike Adams on an interview. And he said, yeah, but we know that the sun gives you sunburn, and we never isolate